Hello. Welcome to this Sunday session. Hopefully we're now live. I kind of forgot how to set it up for a second, so I'm hoping it's all working okay. Let me know if you can see and hear me okay. Just using we've got some like comments in the live chat area. Perfect. You can see a thumbs up. Is anybody joining from the UK today? Yeah, I don't know about the rest of the UK, but certainly like London at the moment is in a bit of a heat wave, and yeah, I swear it's like 1,000,000 degrees. I've been like googling how much it costs to get air conditioning installed, but I think it's probably gonna get cold again by next week, so it's probably not worthwhile. Yeah, I guess shouldn't complain. It's nice. Did anybody sit the paper one of the biology last week? Any A2 students here? And if so, how did you find the paper? Obviously different exam boards have different papers, but what did you guys think? I've only heard feedback from the AQA paper so far, but the people I spoke to, they said they thought it was like yeah, it went pretty well. Nice. OCR was also good. Yeah. Perfect. Yeah, it's nice when you come away from an exam and you think it's gone okay, like not a total disaster. That's always a good thing. Anyway, yeah, so let's jump into what we're doing today. So we're going to go through some stuff on the kidney. So it's a little bit of a tricky topic. It's one that people often don't like. So we'll do the structure of the kidney, and then we'll also do the osmoregulation stuff, like maintaining the potential of the blood. It's quite a big class, so I don't know if we'll get through everything, but we'll do our best. Yeah, a little bit about me. My name's Alex. I've been tutoring and teaching biology for the last 6 years now. Yeah, and yeah, let's get started with this session. So these are obviously live, as you can see. So the reason we do these live is so we can make them as interactive as possible. So yeah, I'm going to chuck questions out. I would recommend you try replying. You can put answers in the chat section, and I can look at what you're saying and try and pick out any like misconceptions, that sort of thing. So you'll get more out of it if you actually the more you engage with it. But obviously, totally optional if you want to. So anyway, yeah. So first question. This isn't in all specs actually. So I don't think everyone would know. For example, it's not in the AQA syllabus. So how is a high glucose concentration detected? There's kind of like a multiple step pathway for this. They swapped around the ascending and descending limbs of the loop of Henle, as in like they made a mistake in the paper, or it was like some weird question. Because they do make mistakes every now and again, even though they obviously proofread it lots of times. They still kind of sneak in sometimes. I'll start writing an answer to this one as well in case people are doing it. Glucose goes into cells. ATP is produced because the glucose is respired. This closes potassium channels. This changes the charge. So voltage change. Oops. What's happened here? There we go. I can't really write in the corner very well. Voltage change. And now calcium channels open. Oh, we can't write here. And then just like in the synapses or in the synapse topic, calcium can move vesicles to fuse with the membrane. So the calcium causes exocytosis. of vesicles. Yeah, which will then release insulin. Yeah, so like I said, not all specs do that. So if you have never heard of that before, chances are it's not in your syllabus. Anyway, yeah, it's quite a long question to be in this. We normally like shorter questions in this part of the session. Okay, so what is the response of the body to a low glucose concentration? Oh, okay, it's just difficult to see where the PCT was. Yeah, okay, I see. Yeah. Nice, Rafaela. Exactly. They're gonna release insulin. Oh, sorry, low glucose concentration. No, you're not going to release insulin. That would actually further exacerbate the problem. What's the hormone you're going to release if you've got low blood sugar? Insulin would be for high blood sugar. Yeah, that's glucagon you want. Your body will also, yeah, which is going to trigger glycogenolysis. And gluconeogenesis. You also get a slight slowing of your metabolism. So if you've got low blood sugar, your body's going to try and find ways to bring that back up. But another thing you can do to help is just lower your general metabolic rate to conserve energy. The flip side, if you've got high blood sugar, your metabolism increases. So I'm sure you guys have all heard of like a sugar high as an expression, but it is actually a thing. You do sort of get a boost of energy from having more sugar or like more blood sugar. So yeah, there is like some logic to that. The respiration rate probably would increase slightly because you're going to need to. Do you mean like? Oh, I was thinking of like respiratory rate as in like how much you're breathing, like ventilation rate. But yeah, the overall rate of respiration in all the cells is higher. Yeah, which then consequently you're going to need to like breathe more to fuel all that extra respiration. Yeah, it's not a huge amount, but it's like a measurable increase. Cool. This is kind of getting a bit messy. Oh well. So how does the binding of adrenaline cause chemical changes within cells? What would we say for this? Again, this could be quite a long answer, so maybe let's try and shorten it. I won't go through the whole pathway, but there's a name for like how this action works. Because if you remember, adrenaline is peptide hormone. This means it's made of protein rather than a steroid hormone, which is made of lipid. So peptide hormones and steroid hormones behave differently due to their different properties. Lipid ones can go straight through the membrane. Peptide ones can't go through the membrane. So generally, they bind to a receptor on the outside, and then this causes a change on the inside, leading to another chemical being released on the inside, and we call that the second messenger. So this adrenaline works through the second messenger model. Again, I think that might not be in all specs. Some specs do more on it than others, and there's a slight difference. Like I think OCR, you guys have to know about G proteins in this like multi-step pathway, whereas AQA don't, and I'm not sure where Excel whether you have them or not. But yeah, second messenger model, and the second messenger in this case is something called cyclic AMP, or just like CAMP for short. Should actually be a little C. like that. So hopefully that's ringing a bell. Okay, this is also another long question. I feel like these are all a lot of these are quite long for this bit. So 1 in 6,000 people have a recessive disease. How many carriers are there in a population of 500? So you're going to need to do a Hardy-Weinberg calculation here. So you need the two equations. So you do actually have to memorize these. So it's q squared plus 2pq plus, oh, sorry, well, it should be, I guess it doesn't really matter. You normally do p squared first, but the equation is still the same. Equals 1, and p plus q equals 1. If you remember, this top one is about genotypes, and this bottom one is about allele frequency in the population. So the way these normally work is you're given q squared. You're basically told like how many show the recessive phenotype. So we're saying 1 over 6,000 is equal to q squared. Once you've got q squared, you square root it. So let's square root 1 over 6,000 to get q. And now if we've got q, we want to get to p. So we do 1 minus this answer, which would then give you q. So this sort of like equation here, that would give you the value for, sorry, that's going to give you the value for p. And now you've got a value for q, and you've got a value for p. You can just times those together. 2 times p times q. And that's going to equal your number of carriers, because the carriers are the heterozygous ones. So 2pq, this is heterozygous. And that's how we calculate it. So yeah, hope that is all clear. If you want to have a go and see if you get the correct answer, you should come out with 13 with like rounded up. So yeah, maybe have a go if you wanted to practice a Hardy-Weinberg. Okay, and then next question: what is the effect of crossing over of linked genes? What would you say for that? Oh yeah, and apologies for anybody who was in the web class a couple of weeks ago that it got cut off after about 20 minutes. My Wi-Fi just went down for like 2 hours, so I tried to like get back on, but it was obviously not happening. I tried to tether my phone as well, but yeah, my like phone signal isn't like very good around here, so that wasn't an option. So apologies if you were cut off there. So again, bit of a vague question, but you can say something like alleles, different combinations of alleles can be inherited together. Inherited. Cool, nice. So yeah, let's jump in to the kidney cell. So we're going to look at the structure. We're going to look at the functions of the different parts of the nephron, and then we'll do the osmoregulation stuff. Oh, LXLA, you guys don't do the kidney at all. So if you're LXLA, I would probably say you can tune out, go do something else that's going to be more beneficial. Obviously, you're welcome to stay, but it's not going to be in your exam. Okay, so the overall structure of the kidney, we can split into 3 parts. We've got the cortex, we've got the medulla, and we've got the pelvis. And these contain different parts of the nephron which perform different functions. So you can see this bit in white here. this is like a model of a nephron, but it's obviously not to scale. Like you have millions of nephrons inside the kidney, so they're really, really tiny. You wouldn't be able to see it like that. But that's just kind of like where the stuff would be proportioned. So you can see the cortex, the outermost layer. This contains the glomerulus, the Bowman's capsule, and both the PCT, proximal convoluted tubule, plus the distal convoluted tubule in here. The medulla contains the loop of Henle and also the collecting duct. And the pelvis pretty much just contains collecting ducts. And the pelvis is basically where all the waste products that have been filtered out of the blood, they drain into this tube called the ureter. So this would then go on to the bladder. So the waste is what we call urine, and it's obviously mostly water, and then you've got various other things like urea, salts, other metabolic waste products that the body doesn't need. So the kidneys are sort of like a filter, and they remove metabolic waste from the blood. Yeah, they're slightly unusual as organs go, insofar as we've got two kidneys. You can actually run perfectly fine with just one kidney. So it's kind of weird for nature to like give us but it's not like a spare. But yeah, like usually stuff that we don't need gets like sort of removed by evolution. So yeah, it is preferable to have two kidneys, but you can definitely get by fine with just one. You have to be a little bit more careful with like what your lifestyle to make some changes. But yeah, there's not many other organs you can do like so well like having only one. Why are you doing the kidney? That's paper one. So it's different for every exam board, Zach. So which exam board are you? Because for AQA, it's like in papers 2 and 3. So yeah, that's slightly unfortunate if it's in just paper 1 for you. But yeah. Is it not in your paper 3 as well? Is that like a mixture of all the topics? I can't remember how the OCR exams are structured now. Is paper 3 like a general one on everything, or is it? You're definitely not going to do kidney again. Because if that's the case, I would say yeah, probably it is on paper 3. Okay, nice. So you can do it. Okay, cool. Yeah. So yeah, some stuff on the kidney might come up. Did the kidney come up on paper 1? Because I guess if there was a lot of questions on the kidney in paper 1, less likely to be in paper 3. But yeah. Cool. Yeah. If you, we've obviously got past recordings. So if you wanted to do a different subject, yeah, you can go back in time. It's obviously not live, but yeah, we've got hundreds and hundreds of sessions on like all the different topics. So if you think one's going to be more beneficial for your exam on Wednesday, then by all means do that. Is it Wednesday or is it Thursday? I can't remember the next paper. Anyway, cool. Yeah, let's carry on with the kidney stuff. So yeah, hopefully that's all good so far, all making sense. And let's try some past paper questions. So fairly straightforward, this first one. Name region P and name structure Q. So what do you say for this? Ouija. Yeah, Welsh board. That's not a super common one. Yeah, fair. Yeah, nice. So P is the cortex, and Q is the ureter. Oh yeah, so not the pelvis. Pelvis would be sort of, I'd say pelvis stops there more or less, and then it becomes the ureter. So as once it's out of the kidney, it's pretty much like ureter by that point. Whereas like that would be pelvis, and this would be the medulla. They've actually drawn them a bit more realistically in this diagram because they're actually these sort of triangle shapes. These are called renal pyramids. But I don't think many people need that. Okay, and then now we've actually got a micrograph. So a photo of a microscope, sort of down the microscope view, and you've got to identify these structures labeled E and F. So I think F is more straightforward. E is pretty hard to tell what it is, but I guess you've got to look at like where this is. So this is in the cortex. So hopefully that'll help. Yeah, of course. Welsh board is common in Wales. Yeah, but what is that like? Is it like 2 million of you guys? Not that many in the grand scheme of things. I think AQA is the most common, and then I think OCR is next, and then LXL, and then Welsh board would be like probably like number 4. The loop of Henle is in the medulla, exactly. So it can't be the loop of Henle, but the loop of Henle would look quite similar, to be fair. They would all the like tubules would look quite samey. But because it's in the cortex, it has to either be the PCT or the DCT. They accepted either for your answer. And then what is F? As a clue, there's another one over here. This bit in the middle, they've labeled is the glomerulus. So therefore, what is this bit around the outside of that? Yeah, it must be the Bowman's capsule. Nice. So it's always a bit harder looking at an actual micrograph image rather than a diagram, because diagrams have been simplified to identify features. So like you can see this, they've simplified it. They've tried to highlight the different features, and that's what you have to do when you're drawing a biological diagram. You're trying to simplify, make it easier to see the different areas and what's going on. Yeah, so it's always tricky when they haven't done that. Anyway, cool. So let's look at some of the kidney functions. So there's 3 main things the kidney, well, sort of 3 stages to what the kidney does. The first part, which happens in the Bowman's capsule and the glomerulus, is ultrafiltration. This is basically pushing out a lot of the substances from the blood. Stuff is removed. So the stuff that passes out, actually we might have a separate slide on this. Well, we can put it here. So what sort of stuff is going to be filtered out from the blood? But the most common thing is water. But what else would we find in there as well? I don't necessarily put everything, but we'll put some of the things. Proteins. Actually, proteins, the plasma proteins, these are the big proteins, they actually stay in the blood. But amino acids would be filtered out for sure. Glucose, hormones, ions, what else could we say? Oh, urea, obviously, is filtered out. And then the next stage, which occurs primarily in the PCT and the DCT, and to a lesser extent the loop of Henle as well, is selective reabsorption. So stuff is filtered out, sort of like a lot of stuff is filtered out, and then we reabsorb the things that we want. So we're selective in taking back the things that we want. Anything that we don't want, we're trying to get rid of, is not reabsorbed, because once it's been filtered out and the substances are in the tubules, they're destined to be excreted from the body. They're going to end up as urine and they're going to go to the bladder. So if you want to keep something, you have to reabsorb it. Anything that's not reabsorbed is going to be excreted. So we take back the things we want. Everything else is then lost as urine. So we can get rid of it from the body. And the final thing the kidney does is it plays a role in osmoregulation. So like water, let's just put osmoregulation. Essentially like maintaining water potential of the blood. So let's go through some of the stages. So let's take a closer look at ultrafiltration. So blood is filtered out of the glomerulus to form, you can call it just filtrate, or you can call it glomerular filtrate. The glomerulus is basically like a little ball of capillaries, and the blood flows into the glomerulus through something called the afferent arteriole, and it comes out of the glomerulus through something called the efferent arteriole. These are actually a different diameter. This is a larger diameter, and this one has a narrow diameter. So the afferent's wider, the efferent's narrower. This creates a bottlenecking effect. So you've got more blood coming in a wider tube, going down to a narrow tube, like a bottleneck. This helps to create a very high hydrostatic pressure. So we have hydrostatic pressure in our blood. That's how the blood moves around the body. It's created by the heart pumping the blood, pressurizing it, creating this hydrostatic pressure. The hydrostatic pressure by itself would already be enough to do some filtering out of the blood. And that happens in the capillary beds normally. That's how we make tissue fluid. But for ultrafiltration, we want to filter out even more stuff. So the bottlenecking effect here creates an extra high hydrostatic pressure in the glomerulus. So we get lots of filtering. So we can add that to this first note. Let's just put afferent arteriole wider. Efferent arteriole creates high hydrostatic pressure. Small soluble molecules are filtered out. And passed into nephron, out of blood. The stuff that's left behind are red blood cells and the plasma proteins. Left behind. They're basically too big to leave through the walls of the capillary, so they stay behind. And there are three things which are sort of doing the filtering. So to get out the blood, you need to move through the actual wall of the capillary itself. You need to move through the basement membrane, which is basically like connective tissue between the cells. And you need to squeeze through these specialized cells called podocytes, which are sort of like shaped a little bit like a mesh, like a net. So yeah, the three of those, capillary endothelium, that's just basically the name for the wall. Basement membrane, oops, weird M, and podocytes, the filtering, or acts as the filter, or act as the mesh, whatever you want to call it. And then the glomerular filtrate is then going to pass in to the PCT. So you can see here, this is the start of the proximal convoluted tubule, where it's then going to undergo selective reabsorption. So, and that's on to our next page. So, selective reabsorption, useful substances are reabsorbed in the PCT. So we're going to take back things like glucose, amino acids, some of the ions. Although if we've got a surplus of ions, we're also going to remove some of them. But some ions will be taken back, and stuff like hormones will be reabsorbed. And as he said, anything that's not reabsorbed is going to be released as waste. So that's how stuff is removed from the body. And we only really talk about urea for A level, and that, to be fair, that is the biggest, like that's the main waste product that you find in urine. Urea actually comes from the breakdown of proteins. So if you use proteins as a respiratory substrate, you're basically respiring protein rather than carbohydrate or lipid. Then you need to remove the ammonia part. If you remember the structure of an amino acid, we'll recap on biomolecules. Looks like this. R, H, and then the amino group. So to respire something aerobically, you produce CO2 plus water. So the rest of the molecule can be turned into CO2 plus water because it's only carbons, hydrogens, and oxygens. But this amino group is a problem. So to be able to respire a protein, you need to chop that off. But if you just leave it as it is, it will form ammonia, NH3, or if it's in water, it will form ammonium ions, NH4+, which are quite toxic, or very toxic, in fact. So what we do is we combine that with CO2 to form urea, which is like a less toxic version of ammonia. And then we still need to get it out of the body. So we've detoxified it, or made it less harmful, but then we actually, excrete it. And that's what gives urine, well, its name and its like distinctive like smell and color. That's urea. That's the like what urea smells like and looks like. I'm not sure which came first, whether the name urea or the name urine. Obviously they're like linked, whether urea is named after urine or whether urine is named after urea. I suspect probably that way around, that urea was the chemical first and then urine was the substance that contained it, but yeah, I'm not exactly sure. definitely not relevant for your exams though. So yeah, not something to worry about. They're not going to ask you that. So yeah, the useful stuff is reabsorbed. Some specs, I think it might just be AQA, link this to co-transport. And you have to like describe the mechanism using sodium ions, and like active transport of sodium sets up a concentration gradient, which then drives the facilitated diffusion of the glucose and amino acids. But because we've got a lot to get through, I think let's not get into that. Let's not get into that now. Let's add a note. That's an indirect form of active transport. Cool. Nice. So let's look at the water uptake part of this now. So yeah, it's always a little bit tricky to know which part we should start with here. So yeah, I'm going to do it in this order, starting with the ascending limb of the loop of Henle. So we say that something like ions pumped out of ascending limb of loop of Henle using active transport. The descending limb and the ascending limb is impermeable to water. The descending limb, this is permeable to water. So we've pumped out all these ions. This creates a low water potential in the medulla, because if you pump out ions, ions are going to lower, like any solute is going to lower the water potential of the surrounding area. So they lower the water potential. This creates a water potential gradient. And so water moves out of descending limb via osmosis, or like down the water potential gradient. Yeah, which then creates a low water potential concentration in the tubule. Because you now got a low water potential in the tubule because all the water's moved out, you actually end up with ions moving out at the bottom of the loop of Henle, which is an example of a countercurrent multiplier effect. Some specs don't talk about that as much, but it's something you could add in. Yeah. Anyway, water potential of filtrate is very low by bottom of loop of Henle. So ions move out. Yeah, then water plus ions are reabsorbed in the DCT. And this is actually where the ions are fine-tuned. So another part of osmoregulation is absorbing water and ions in the correct amount. And yeah, we just say like water reabsorbed in the collecting duct. Cool. Nice. So yeah, hopefully that's making sense. Let's try some past paper questions on that stuff that we've gone through. So, got a diagram here, got some labels. The first part, kind of easy to miss this question. 3 marks, just labeling, just naming L, M, and N. So what are these 3 things? So L, have you recognized is the glomerulus? M is the Bowman's capsule, and N is the PCT. You shouldn't really use, if you're writing this in an exam, you shouldn't write PCT, or you definitely shouldn't write BC for Bowman's capsule. You should actually write it out in full. I'm just writing it to save some time, but just to make sure you guys are aware that you should write that properly. Okay, then we've got 5 statements here that match up to a part of the nephron. So I'll number them in case you wanted to use the numbers in your answers. So what name, like you can just name the part of the nephron that matches up with those statements. So what do you guys say for these things? I swear it's getting hotter. There was like a little breeze earlier, but it seems to have stopped. So walls impermeable to water. This is the loop of Henle. More specifically, this is the ascending limb. Of the loop of Henle. Glucose is reabsorbed into the blood, is of course the PCT. ADH acts on the walls. We haven't really talked about ADH yet. We're going to talk about it on the next page. It stands for antidiuretic hormone, but this acts on the walls of the collecting duct. Podocytes are, of course, in the Bowman's capsule. And most of the water is reabsorbed into the blood. This one always catches people out. A lot of people would say the collecting duct, but that would actually be incorrect. Most of the water, believe it or not, is actually reabsorbed in the PCT. So even before we get to the loop of Henle, around 85% of the water has already been reabsorbed back into the blood. Which, if you think about it, makes sense, because we've been doing active transport of all of the like glucose and amino acids. A lot of the solute has come out. So water, we've pumped out the solute, water will follow now via osmosis. So yeah, we've only got around 15% of the original water by the time we even get to the loop of Henle. In the PCT. Yeah, and then the actual like fine tuning of the water happens, the water concentration happens in the rest of the kidney. Cool, nice. So we've got now like more like a longer answer question. I mean, still not a very long answer. It's only 2 marks. So have a read of the question. And I'll give you guys like a little bit of time to think about it, and then I'll write out like what would be a suitable answer for that question. So longer loops are handy. How do they help them survive in the desert conditions? We can say something like longer loops equals more salt or more ions or more sodium pumped out of ascending limb. This creates a greater water potential gradient between the medulla and the filtrate. Therefore, more water is reabsorbed. And this is what allows the kangaroo rat to live in the desert, and what it's a way of conserving water. So you'll find that animals that live in very dry conditions, they tend to have these very long loops of Henle, which allow them to reabsorb a lot more of the water. Some animals are really good at this. So snakes, for example, snakes can reabsorb so much water that their urine is essentially solid. They just sort of secrete solid urea because they're conserving like so much of the water. So depending on the conditions, that may be a useful adaptation. If you live in an environment where water is pretty plentiful, then that's not something you'd need. It's just like it's a waste of energy, essentially. So that's why only things that need it would have developed that mechanism. Because the active transport of the sodium ions is using up energy. So you wouldn't do it unless you needed to. Okay, cool. So we've got around 10 minutes now to quickly go through osmoregulation, which should be just about enough time. So I think, we should actually be on track to get everything done. So osmoregulation is controlled in the hypothalamus by these things called osmoreceptors, located in the hypothalamus. And they can detect low water potential. If they detect low water potential, they will release ADH, antidiuretic hormone, which will travel via the bloodstream and it will act on the walls of the collecting duct. We'll look at a little bit about how that works on the next page. Actually, I think we're doing it on this page. So ADH travels via the bloodstream, it binds the walls of the collecting duct and it makes them more permeable. So makes walls more permeable. Therefore, more water reabsorbed. And this increases the water potential of the blood. So you release ADH when you want to conserve water, to raise the water potential of the blood. So you're going to minimize how much water you're losing as urine. And basically conserving it. If you're hydrated, then it's the opposite. So you're going to release less ADH. The osmoreceptors will detect a high water potential, stop releasing ADH, and then the walls of the collecting duct become less permeable, and therefore more of the water is going to end up in the urine, and you create dilute urine that's removing the water. So this is all going on, like in your body. Obviously, it's not under conscious control. It just yeah, one of the homeostatic mechanisms that just sort of ticks along in the background. Cool. So that's the mechanism. Let's have a look at some past paper questions. So yeah, there's some context here. You should always read context when you're doing a question. In this case, you probably could get away with not reading it, but I'll give you like 20 seconds, have a look, and then we'll go through the questions. The first one was just a nice, easy one marker. You just have to name the gland that releases ADH. Yeah, nice. So that's the pituitary gland. Perfect. And explain how the structure of protein molecules allows them to form channels through which only water molecules can pass. So this isn't really a question so much on the kidney specifically. It's more like a protein structure question. I hope that's a bit of a clue, but how you should actually set out your answer. So how does the structure of the protein molecules allow them to form channels through which only water can pass? Yeah, I'll give you guys a second to have a go at that. I'll write out what could be like a good model answer. Yeah, say something like proteins have a specific, proteins have a tertiary structure. You could probably get away with 3D structure, but tertiary is better. Tertiary proteins is specific for each, or specific in this case for the water molecules. What is specific? Let's put has a specific shape. Which fits water. Cool. Yeah, the channels are called aquaporins, exactly. Yeah, nice. So final question. Explain how the cells of the collecting duct are able to absorb water from the filtrate through protein channels in their plasma membranes. Again, 2 marks. So how come water will move through the aquaporins, basically, is what they're asking. Not active transport, it is passive. Yeah, and to do passive transport, you therefore need to have a concentration gradient. And in the case of water, we call that concentration gradient a water potential gradient. Yeah, so it's osmosis, exactly. So yeah, you actually can't actively transport water. You have to you can actually transport the ions, which then allow for the concentration gradient. You can't like pump water directly. So yeah, something like more negative water potential. In tubules than infiltrate. And that was actually all set up by pumping out the ions in the loop of Henle. So that's why you do the pumping out the ions, or one of the reasons. So more negative water potential in the tubules and the filtrate, therefore water moves down water potential gradient, or like moves via osmosis into tubules, something like that, or eventually into the blood, but tubules first, and then into the medulla, and then into the blood. Yeah. So yeah, that would be like a suitable answer to that. Two marks, one for each of those statements. And I think that's that. So that was quite a quick run through the kidney. It is a big topic. It's probably like too much to really try and do in one session. So it might be a little bit rushed. There's a few more things you could look through on that. Why does collecting duct need aquaporins? Oh, so the osmosis is occurring through the aquaporins. So yeah, good question. So intertubules via aquaporins. You don't need it for this question, but that's how it's actually being reabsorbed by osmosis. Water can go through membrane a little bit, but because it's hydrophilic, it doesn't go very well through. So it kind of has to go through channels to move at any sort of decent speed. Yeah, so that's that. I think, if you wanted to, we did these seminars over the Easter holidays, which obviously they're done now, so you can't attend them. But we're selling the recordings. So yeah, these are obviously like super helpful for your exams. So if you did want to like download those, you can go to our website. There's like tips on exam technique. We go through some of the trickier topics and just like break down the main things you need to know. And you get this like booklet sent to you, which is kind of like, it's got lots of good tips, kind of like a cheat sheet for the different topics and like practice questions, that sort of stuff. And we got them in the 3 sciences and maths. So maybe some of you guys went to these seminars or you attended them online. If not, you can subscribe to those. And this kind of like, this is the stuff we do on the exam technique stuff, looking at the command words, breaking down how you should approach a question, and then how you should structure your answers to maximize marks. The trickier topics obviously is just like what we've identified as ones that students find tricky. So we do like inheritance, we do photosynthesis, respiration, we do, what else is in there? Some of the maths questions like the stats. I know a lot of people don't like that. And again, how to like structure answers for that. So yeah, you can go to this link and you can access those. Anyway, I will end there. Oh, pretty much bang on 5, so that's perfect. Good luck for paper 2 for anybody who's sitting that next week. Hope it all goes well. And probably after you've done paper 2, you're pretty close to you've probably done the majority of your exams. So the end is in sight. And you got paper 3 obviously for bio the week after, but it's probably your schedule is getting a little bit more chilled after next week. So you're nearly there. Keep going, and hopefully goes well. Cool. Yeah. So see you guys in the next session. I think we probably actually don't have another YouTube class now till after your exams. So if you're going to join for that one, it'll be at some point in the summer. If you're an A2 student, you're probably not gonna be there, so which is totally fair enough. But see you guys then.